So the last formed element we're going to take a look at are thrombocytes. So thrombocytes are platelets, and platelets are really are just fragments of cells. They're not a true cell, and their job is blood clotting. So the process of platelet formation is thrombopoiesis, and it's going to be regulated by a hormone called thrombopoietin. Okay. So basically what happens, here you go, you're starting with the same stem cell, a hemocytoblast in bone marrow. It's going to receive a chemical signal from the thrombopoietin, and it's going to enter into a developmental pathway. I don't really care that you learned the different names. The one that I do want you to know is a megakaryocyte. So a megakaryocyte is the last stage that a platelet is a complete cell. So the platelets are they're a result of this megakaryocyte kind of exploding and forming all these little fragments of a cell. The granules that are found in the individual platelets help with the clotting process. The clotting process is referred to as hemostasis. So don't get that confused with homeostasis. It's hemostasis. Okay, so platelets are responsible for blood clot formation, and their job is to prevent blood loss due to a torn blood vessel wall. It's a very quick, localized, and carefully controlled process. So you have 3 phases that occur in rapid sequence. The first one is vascular spasm, the second is platelet plug formation, and the last one is coagulation. So for vascular spasm, so if you look here, here's a damaged blood vessel wall. You're going to have vascular spasm occurring first. First thing that happens is vasoconstriction. So the smooth muscle is going to vasoconstrict, and it's basically to just reduce the overall surface area of exposure or cut blood vessel wall. So you're constricting, and it's also to prevent blood from losing so much blood, all this blood seeping out. So what causes this vasoconstriction is a chemical that's released by the simple squamous cells of the damaged blood vessel, and also by the platelets themselves that are present there. So the blood vessel is going to constrict so that the opening in the blood vessel wall becomes smaller. It reduces bleeding, and then that helps the further actions of the platelet to secure the site or have homeostasis. After the vasoconstriction, you're also going to have collagen fibers that are going to be exposed as a result. So these collagen fibers are going to be here, and they provide a surface for the platelets to adhere to when they're going to form their plug, which is the next step. So step 2 is platelet plug formation. The platelets are going to form a plug that's going to seal that break in the blood vessel wall. The exposed collagen fibers are going to attract platelets, and then when the platelets adhere to these collagen fibers, they become sticky, they develop pseudopods, which are kind of like feet-like projections, and they release a chemical. And this chemical then attracts other platelets to that site. And then those platelets come, and then they attach to the collagen fibers, they develop pseudopods, they become sticky, and then they release a chemical. And then more platelets come, and the same thing. The platelets become sticky, they develop pseudopods, and they make a platelet plug. This is a positive feedback mechanism. The formation of this platelet plug is a positive feedback cycle. So if you remember what you know about a positive feedback cycle, once you have the initial stimulus, it moves forward in the same direction, so it's exaggerated or enhanced, and that's what you see here. So the first platelets there release a chemical that causes the platelets to become sticky, they develop pseudopods, they stick to the collagen fibers, and then they release a chemical, and that attracts more platelets, and they become sticky, develop pseudopods, and then they release a chemical. So you keep having that positive feedback mechanism continuing until the platelet plug is formed. Once the platelet plug is formed, the next step is coagulation. So coagulation is basically transforming the blood from a liquid state to a gel-like consistency. Under normal conditions, you have anticoagulants, which are chemicals in the blood that prevent blood clotting. But when this clotting process begins, then procoagulants dominate. So it's through a series of what we call intrinsic and extrinsic pathways that are basically going to convert prothrombin into thrombin to fibrinogen, then to fibrin. Okay, so you have two different processes that are occurring at the same time. You have the intrinsic pathway, and what you want to know about the intrinsic pathway is that all the components for this process of converting the blood from liquid to gel are found in the blood in an inactive state, and they become activated when the platelets release chemicals to activate this process, to get coagulation occurring. So the chemicals are going to release the platelets are going to release a chemical, and it's going to stimulate the production of procoagulants, and then you're going to have this intrinsic pathway occurring that's basically going to convert prothrombin into fibrin. As far as the extrinsic pathway, it's happening at the same time. Extrinsic pathway, extrinsic, it means it's outside of the blood. So this is what's going to happen. The same process, you're going to have chemicals released by the damaged tissue cells. So it's not in the blood, it's the damaged tissue cells that release a chemical that's going to convert prothrombin. It basically becomes prothrombin becomes thrombin, and then thrombin becomes fibrinogen, and then what you want to end up with is fibrin. And the fibrin is going to form this mesh, and it's going to kind of help to glue the platelets together. So this is showing you a fibrin mesh that's created as a result of the prothrombin being converted into fibrin. So you can see all these string-like things. Those are the fibrin threads. It's going to trap red blood cells and other formed elements. So it kind of forms this mesh, and it helps to form a clot. So usually you're going to have a clot formed within 3 to 6 minutes of the initial injury. There are like 13 clotting factories. Most of them are going to be synthesized in the liver. You can read that in the textbook if you want. I don't really care to get into that. You just have to know that the clotting factors help to contribute to the intrinsic and the extrinsic pathway, and to get. It's like this big, long 15-step process, and you're basically converting the prothrombin into fibrin. So that's all I care that you know. Last thing that happens is clot retraction and repair. So the platelets contain actin and myosin, which are contractile proteins. So they're going to contract and pull on the fibrin strands, and what you end up as a result is a compacted clot. This also decreases the size of the injured area to make it easier for fibroblasts, smooth muscle cells, and other endothelial cells to repair the damage. You have platelet-derived growth factors that are released by the platelets, and they stimulate the smooth muscle and the fibroblasts to rebuild the wall, and the endothelial cells to repair the lining of the vessel. Once the healing has occurred, the clot has to be removed, and you have chemicals that are going to activate plasminogen, which is going to produce plasmin, and plasmin is an enzyme that basically digests and dissolves the clot. So it's happening from the inside out. And there you go. There's your clotting process.